Okay, Elena, I need you to explain something to me. We've spent decades looking for Earth 2.0, a nice, rocky planet with blue oceans and fluffy clouds. And now you're telling me the best candidate for alien life is a planet that's 8 times heavier than Earth, covered in a global ocean, wrapped in hydrogen gas, and orbiting a dim red star?
That's actually a pretty accurate summary.
And scientists found what might be the chemical signature of life there, in a place that shouldn't be able to support life at all?
Welcome to the paradox of K2-18b. It breaks every rule we thought we knew about habitable planets, and yet here we are debating whether something is alive in its oceans.
You know what this sounds like? It sounds like the universe looked at our textbooks on planetary habitability and said, "That's adorable. Watch this."
That's actually a perfect way to put it. K2-18b is forcing us to completely rethink what habitable means, and the story gets wilder the deeper you go.
All right, folks, welcome to the planet 124 light-years away that shouldn't support life. I'm Marcus Chen.
And I'm Dr. Elena Vasquez. Today, we're diving into one of the most controversial and exciting exoplanet discoveries in history, a world that might be teeming with alien microbes or might just be tricking us with chemistry we don't understand yet.
We've got biosignatures, ocean worlds, hydrogen atmospheres, and scientists arguing with each other in peer-reviewed journals. It's going to be a wild ride.
And fair warning, by the end of this, you might have more questions than answers. But that's kind of the point.
The best science always leaves you wanting more. Let's get into it. All right, let's start at the beginning. K2-18b. That's not exactly a catchy name. Where does it come from?
The name comes from the K2 mission, which was basically Kepler's second act. The original Kepler Space Telescope had this incredible run, discovering thousands of exoplanets. But in 2013, it lost 2 of its 4 reaction wheels. These are basically gyroscopes that keep the telescope pointed precisely.
So the telescope broke?
Partially. NASA engineers are incredibly creative, so they figured out a way to use solar radiation pressure to stabilize the telescope. They called this new mission K2, or Second Light. It wasn't as precise as the original Kepler, but it could still find planets.
And K2-18 is the star.
Exactly. K2-18 is a red dwarf star in the constellation Leo, about 124 light-years from Earth. The suffix B means it's the first planet discovered orbiting that star. There's actually a second planet, K2-18c, but that one's much closer to the star and not in the habitable zone.
So when was K2-18b actually discovered?
The discovery was announced in 2015 by a team led by Benjamin Montet. They spotted the telltale dip in starlight that happens when a planet crosses in front of its star, what we call a transit. But at the time, it was just another interesting exoplanet among thousands.
What made it special?
Location, location, location. K2-18b orbits in its star's habitable zone, the region where temperatures could potentially allow liquid water to exist on a planet's surface. And here's the thing about red dwarfs. They're much cooler and dimmer than our sun, so their habitable zones are much closer in.
How close are we talking?
K2-18b orbits at about 21 million kilometers from its star, with an orbital period of roughly 33 days. Compare that to Earth, which orbits at 150 million kilometers with a 365-day year. So K2-18b is much closer to its star, but it receives about the same amount of energy that Earth gets from the sun.
That's actually kind of perfect, isn't it?
On paper, yes. The planet receives about 1,368 watts per square meter of stellar radiation, which is remarkably similar to Earth's insolation. If you removed the atmosphere from the equation, K2-18b would have a temperature somewhere between -23 and +27 degrees Celsius.
That's literally t-shirt weather to Antarctic expedition weather. That's a livable range.
Right, but here's where things get complicated. K2-18b is nothing like Earth. It's 8.6 times Earth's mass and 2.6 times Earth's radius. That puts it in a category we call sub-Neptunes, planets bigger than Earth but smaller than Neptune.
And we don't have any sub-Neptunes in our solar system.
Exactly. There's this weird gap in our solar system between Earth and Neptune. We've got rocky planets up to Earth's size, and then we jump to ice giants that are 4 times Earth's radius. Sub-Neptunes fill that gap, and they're actually the most common type of planet in the galaxy.
So the most common type of planet in the universe is something we've never seen up close?
Welcome to Exoplanet Science. We're constantly discovering that our solar system is actually kind of weird compared to most planetary systems out there.
Okay, so K2-18b is discovered in 2015. It's in the habitable zone, it's a sub-Neptune, but there are probably thousands of planets that fit that description. What elevated it to poster child status?
2 words: water vapor. In 2019, 2 independent teams using the Hubble Space Telescope announced they'd detected water vapor in K2-18b's atmosphere. This was huge, the first time water had been detected in the atmosphere of a habitable zone exoplanet that wasn't a hot Jupiter.
Hot Jupiters being those gas giants that orbit really close to their stars.
Right. We'd found water on other exoplanets before, but they were all in scorching hot orbits. K2-18b was different. It was cool enough that water could potentially exist as liquid.
Who made the announcement?
There were actually 2 papers published almost simultaneously. One was led by Angelos Tiras at University College London, and the other by Bjorn Benneke at University of Montreal. Tiras famously said, "Finding water on a potentially habitable world other than Earth is incredibly exciting."
I bet the internet went absolutely bonkers.
Oh, it did. Headlines everywhere screamed about finding a second Earth or proof of alien life, which, to be clear, it absolutely was not. The researchers were very careful to say K2-18b is not Earth 2.0. It's significantly heavier and has a completely different atmospheric composition.
But the hype train had left the station.
Full speed ahead. And honestly, that's when the real scientific debate started, because finding water vapor raises more questions than it answers. How much water? In what form? Is there an ocean? Is there a surface at all?
Wait, is there a surface at all? You mean K2-18b might not have a solid surface?
That's one of the big unknowns. Based on its mass and radius, K2-18b has a density of about 2.67 grams per centimeter cubed. Earth's density is 5.51, Neptune's is 1.64. So K2-18b is somewhere in between.
Which tells us
that it almost certainly has a significant volatile component: water, hydrogen, helium, or some combination. But the exact internal structure is degenerate, meaning multiple configurations could explain the data. It could be a mini Neptune with a rocky core, an icy mantle, and a thick hydrogen atmosphere, or it could be what we now call a Hycean world, a water world with a thin hydrogen atmosphere over a global ocean.
And those 2 scenarios have completely different implications for life.
Completely different. A mini Neptune probably doesn't have a habitable surface. The pressure and temperature at depth would be crushing and scorching. But a Hycean world, that might have oceans with conditions similar to Earth's deep sea environments.
So everything hinges on which scenario is correct.
And that's where Niku Madhusudan enters the story. He's an astrophysicist at Cambridge who became absolutely obsessed with K2-18b and the possibility that it represents an entirely new class of habitable worlds.
The Hycean concept was his idea?
He coined the term in 2021, publishing a paper that essentially said, "Hey everyone, we've been looking for Earth twins, but maybe we should be looking for something completely different." He argued that Hycean worlds could be far more common and far easier to study than rocky planets like Earth.
And K2-18b was his poster child.
It was perfect for his hypothesis. The right size, the right temperature, hints of water, and critically, because sub-Neptunes have extended atmospheres, they're much easier to study spectroscopically than rocky planets with thin atmospheres like Earth.
So he basically said, stop looking for another Earth. Start looking for ocean worlds with hydrogen skies.
That's exactly what he said. And in 2021, it was considered intriguing but speculative. Fast forward to September 2023, and the James Webb Space Telescope turned that speculation into front-page news.
Okay, I need you to explain the Hycean concept properly, because I think this is where a lot of people get confused. What exactly is a Hycean world?
The name is a portmanteau. Hy from hydrogen and Cean from ocean. A Hycean planet is defined by two key features: a hydrogen-rich atmosphere overlying a liquid water ocean. The ocean could be tens or even hundreds of kilometers deep, sitting on top of a mantle of high-pressure ice, which itself sits on a rocky iron core.
So imagine Earth's oceans, but planet-wide and incredibly deep, with a sky made of hydrogen instead of nitrogen and oxygen.
Exactly. And here's what's revolutionary about this concept. Hycean worlds dramatically expand the habitable zone. Traditional habitability models focus on rocky planets with atmospheres like Earth. But hydrogen is an incredibly effective greenhouse gas. A Hycean planet can maintain liquid water at much greater distances from its star than a rocky planet could.
Because the hydrogen atmosphere traps more heat.
Right. Madhusudan's calculations showed that the habitable zone for Hycean planets extends much further out than the traditional habitable zone. You could have a water world maintaining liquid oceans even if it receives far less stellar radiation than Earth does.
And that means more planets become potential candidates for life.
Many more. The 2021 paper identified several variations of Hycean worlds. There are regular Hyceans with planet-wide habitability. There are dark Hyceans, tidally locked planets where only the permanent night side is cool enough for liquid water, while the day side is scorching. And there are cold Hyceans that receive minimal stellar radiation but maintain liquid oceans through the greenhouse effect.
Wait, could a cold Hycean even be a rogue planet, one without a star?
Theoretically, yes. If the hydrogen atmosphere is thick enough and there's enough internal heat from radioactive decay, a free-floating Hycean world could maintain liquid oceans indefinitely, no star required.
That's genuinely wild. But let me play devil's advocate here. Just because a planet has liquid water doesn't mean it can support life, right?
Absolutely correct. Water is necessary for life as we know it, but it's not sufficient. You also need energy sources, the right chemical building blocks, and some mechanism to cycle nutrients. On Earth, plate tectonics plays a crucial role in maintaining habitability by recycling carbon and other elements.
And Hycean worlds probably don't have plate tectonics.
Almost certainly not. The deep ocean and ice mantle would prevent the kind of crustal dynamics we see on Earth. So if Hycean worlds are habitable, the biological cycles would have to work very differently.
What about the elements needed for life? The whole CHOMPS thing. Carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur.
That's actually one of the interesting challenges. On Earth, many of these elements become biologically available through weathering of silicate rocks. On a Hycean world with a deep ice mantle, that process wouldn't work. The bioessential elements would have to come from other sources: asteroid impacts, precipitation from the primordial atmosphere, or transport through the ice from the rocky core.
So life on a Hycean world would need to figure out a completely different way to get its nutrients.
Potentially. But here's the thing. We know life is incredibly adaptable. Extremophile organisms on Earth thrive in conditions we once thought impossible. Deep-sea hydrothermal vents host entire ecosystems that never see sunlight and live off chemical energy from the Earth's interior.
And a Hycean ocean might have similar environments.
It's possible. If there's any geothermal activity at the ocean floor, where the ice meets the rocky core, you could have chemical energy sources supporting life. The key insight from Madhusudan's work is that we shouldn't dismiss planets just because they don't look like Earth.
Don't look like Earth is putting it mildly.
8 times heavier, hydrogen atmosphere, global ocean with no land. This is about as un-Earth-like as you can get while still having liquid water.
And that's precisely the point. The universe doesn't care about our preconceptions. Life might emerge in places we never imagined because we were too focused on finding Earth twins.
Okay, but there are skeptics of the Hycean hypothesis, right?
Definitely. Some researchers argue that the conditions deep in a Hycean atmosphere might create pressures and temperatures too extreme for the ocean to remain habitable. Others question whether the chemical cycles necessary for life could really operate on such a planet. There's also debate whether K2-18b is actually a Hycean world at all. It might be a mini Neptune with no habitable surface.
How would we distinguish between those scenarios?
That's exactly what the James Webb Space Telescope was designed to help us figure out. By analyzing the light passing through K2-18b's atmosphere during transits, we can identify specific molecules. Different compositions tell different stories about what's happening on the planet.
And that brings us to September 2023.
And that was the day everything changed. Nuku Madhusudan and his team published results from JWST observations that detected methane and carbon dioxide in K2-18b's atmosphere, the first carbon-bearing molecules ever found in a habitable zone exoplanet's atmosphere.
Why is that significant?
Because the combination tells a story. If K2-18b were a mini Neptune with no surface, we'd expect to see lots of ammonia alongside the methane. Both are common in hydrogen-rich atmospheres in chemical equilibrium. But JWST found methane and carbon dioxide with a shortage of ammonia.
And that shortage of ammonia is the key.
It's huge. The absence of ammonia combined with the presence of carbon dioxide is exactly what you'd predict for a Hycean world. If there's an ocean, it would absorb ammonia while carbon dioxide cycles between the ocean and atmosphere. The chemical fingerprint was consistent with Madhusudan's hypothesis.
So the data supported the ocean world interpretation.
It supported it strongly enough to make headlines worldwide. But that wasn't even the most explosive finding, because buried in the same paper was something that really set the scientific community on fire: a tentative detection of dimethyl sulfide.
I'm guessing that's not just another random chemical.
No, Marcus. Dimethyl sulfide, DMS for short, is what we call a potential biosignature. On Earth, it's produced almost exclusively by life. And if it's really there on K2-18b, it could be the strongest hint of alien life ever detected.
That's a hell of a claim.
It is, and that's exactly why the scientific community responded with a mix of excitement and extreme caution.
So Elena, September 2023, the James Webb Space Telescope reveals methane, carbon dioxide, and possibly dimethyl sulfide in K2-18b's atmosphere. Walk me through exactly how they detected this.
It all comes down to a technique called transmission spectroscopy. When K2-18b passes in front of its star from our perspective, a transit, a tiny fraction of the starlight passes through the planet's atmosphere before reaching us. Different molecules absorb light at specific wavelengths, leaving distinctive fingerprints in the spectrum.
So you're essentially looking at the star's light filtered through the planet's atmosphere.
Exactly, and JWST is extraordinarily good at this. The 2023 observations used two instruments: NIRIS, the near-infrared imager and slitless spectrograph, and NIRSpec, the near-infrared spectrograph. Together, they covered wavelengths from 0.8 to 5 microns.
How much telescope time did this require?
Just 2 transits. And here's what's remarkable. One JWST transit observation provided comparable precision to 8 Hubble observations conducted over several years. The upgrade in capability is staggering.
And what did they see?
The spectrum showed clear absorption features from methane and carbon dioxide. Methane has distinctive bands around 1.15, 1.4, 1.7, 2.3, and 3.3 microns. Carbon dioxide shows up around 4.3 microns. Both were detected with high confidence.
What about ammonia?
Notably absent, or at least present only at very low levels below the detection threshold. This was critical because ammonia should be abundant in a hydrogen-rich atmosphere in chemical equilibrium. Its absence suggested something was removing it, possibly dissolution in an ocean.
And then there's the DMS.
Ah, the controversial part. The spectrum showed a potential feature that could be explained by dimethyl sulfide, but the detection significance was low, around 1 to 2 sigma.
For our listeners who don't speak statistics, what does that mean?
In science, we use sigma levels to describe how confident we are that a signal is real and not random noise. A 1 sigma detection means there's about a 32% chance the signal appeared by chance. A 2 sigma detection reduces that to about 5%. The gold standard for a confirmed detection is 5 sigma, which means only a 0.00006% chance of it being a fluke.
So the 2023 DMS detection was nowhere near confirmation.
Nowhere near. Madhusudan was very careful about this. His exact words were, "The inference of DMS is less robust and requires further validation." He wasn't claiming they'd found life, he was saying they'd found something interesting that warranted more observation.
But the headlines...
The headlines were less careful. Signs of life on alien world. JWST discovers potential biosignature. The hype machine went into overdrive.
Was Madhusudan upset about that?
I think he understood the double-edged sword of publicity. On one hand, it brought attention to important work and secured more telescope time. On the other hand, it set expectations that the data couldn't yet support. He kept emphasizing that more observations were needed.
So what happened next?
2 things. First, other research teams started analyzing the same data independently. And second, Madhusudan's team was granted more JWST time to observe K2-18b with a different instrument, MIRI, the mid-infrared instrument, which operates at longer wavelengths between 6 and 12 microns.
Did the independent analyses agree with Madhusudan's findings?
Partially. Everyone agreed on the methane detection. The carbon dioxide detection was confirmed by some teams, but questioned by others. And the DMS hint was extremely controversial. Some analyses found no significant evidence for it at all.
That's not encouraging.
It's science. Independent verification is how we separate real signals from artifacts and wishful thinking. The fact that there was disagreement meant more data was essential.
And the MIRI observations?
They were conducted in April 2024, and the results were announced in April 2025. And they're fascinating, both for what they found and for the debate they've sparked.
All right, hit me with it.
The MIRI data showed a spectral feature in the mid-infrared that's consistent with either dimethyl sulfide or a closely related molecule called dimethyl disulfide, DMDS. The detection significance improved to about 3 sigma.
That's better than before, but still not 5 sigma.
Correct. A 3 sigma detection means there's about a 0.3% chance the signal is noise. It's suggestive, but not definitive. The team estimates that with another 16 to 24 hours of JWST telescope time, they could potentially reach 5 sigma.
So we're tantalizingly close to a confirmed detection.
We're close to confirming that the molecule is there, but here's the crucial caveat. Detecting DMS doesn't automatically mean detecting life. That's a whole separate debate.
Right, because even if DMS is present, it might have non-biological origins.
Exactly. And this is where the scientific community has gotten into some heated arguments. On Earth, DMS is produced almost exclusively by marine organisms, primarily phytoplankton. It's what gives coastal areas that distinctive ocean smell.
And on Earth, there's no known natural abiotic source of atmospheric DMS.
So on Earth, DMS equals life. On Earth, yes, but K2-18b isn't Earth. It has a completely different atmospheric composition, different temperature and pressure conditions, different chemistry. Just because something is only produced by life on Earth doesn't mean it can only be produced by life everywhere.
And there's now evidence of non-biological DMS production.
In October 2024, researchers analyzing data from the Rosetta mission announced they detected DMS on comet 67P Churyumov-Gerasimenko. Comets are definitely not alive. This showed that DMS can form abiotically in at least some space environments.
That's a pretty significant blow to the biosignature argument.
It complicates the picture, certainly. But there's a counter-argument. The DMS concentrations apparently present on K2-18b are enormous compared to Earth, thousands of times higher, over 10 parts per million, compared to less than 1 part per billion on Earth.
And that level is hard to explain without biology.
According to Madhusudan's team, sustaining such high concentrations would be implausible without a significant biogenic flux. In other words, you'd need something continuously producing massive amounts of DMS to maintain those levels in the atmosphere. On Earth, that something is life.
But couldn't there be some exotic chemistry on K2-18b that produces DMS abiotically?
That's exactly what skeptics argue. The conditions on K2-18b are unlike anything we've studied in detail. A hydrogen-rich atmosphere with potential ocean chemistry could create photochemical pathways we haven't considered. Just because we can't imagine a non-biological source doesn't mean one doesn't exist.
So where does the scientific consensus stand right now?
There isn't really a consensus. That's the honest answer. You have one camp that's cautiously excited about the biosignature possibility. You have another camp that thinks we're getting way ahead of ourselves. And you have people in the middle who are calling for more data and more theoretical work on alternative explanations.
What about NASA's position?
In July 2025, a NASA-led reanalysis of all the JWST data was published. They used Bayesian analysis methods and incorporated multiple independent analyses. Their conclusion, the water-rich atmosphere is confirmed, methane and carbon dioxide are present, but there's no conclusive evidence of dimethyl sulfide. The signal exists, but it doesn't meet the threshold for definitive confirmation.
That sounds like a pretty significant pushback.
It is, but it's not a refutation. The NASA team isn't saying DMS isn't there. They're saying we can't be confident it's there based on current data. They also noted that even if DMS is present, abiotic processes could produce it in hydrogen-rich atmospheres like K2-18b's.
So we're in this frustrating middle ground where the data is tantalizing but not conclusive.
Welcome to cutting-edge science. The frustration is part of the process. And honestly, I think Madhusudan said it best. We have entered a new era in the search for life beyond Earth, where it is possible to detect and debate about potential biosignatures. This is unprecedented.
Even if K2-18b doesn't have life, the fact that we can have this debate at all is remarkable.
Exactly. 10 years ago, we couldn't detect individual molecules in the atmosphere of a planet 124 light-years away. Now we're arguing about whether one of those molecules indicates biology. That's incredible progress.
All right, let's really dig into this biosignature question, because I think it's where the rubber meets the road. What exactly makes something a biosignature?
A biosignature is any substance, group of substances, or phenomenon that provides evidence of past or present life. The key word is evidence, not proof. Biosignatures reduce the probability of non-biological explanations, but they rarely eliminate them entirely.
So it's about weighing probabilities?
Exactly. The ideal biosignature is something that is 1. Definitively produced by life, 2. Detectable from a distance, 3. Has no known abiotic sources in the relevant environment, and 4. Would be destroyed quickly without continuous production.
And DMS checks those boxes on Earth?
On Earth, yes. Marine phytoplankton produce a precursor molecule that breaks down into DMS. The DMS is released into the atmosphere where it's quickly destroyed by oxidation. Its lifetime is only a day or two. The only reason there's any DMS in Earth's atmosphere at all is because phytoplankton are constantly making more.
So if we detected DMS on another planet, we'd know something must be actively producing it.
That's the logic. But here's where it gets complicated. The no known abiotic sources criterion is based on our knowledge of Earth chemistry. K2-18b has a hydrogen-rich, reducing atmosphere, the opposite of Earth's oxygen-rich, oxidizing atmosphere. Chemical reactions work very differently in reducing environments.
How differently?
On Earth, DMS gets destroyed quickly because it reacts with oxygen-containing molecules. In a hydrogen-rich atmosphere without much oxygen, DMS might be much more stable. That changes the calculation entirely.
So if K2-18b produces less DMS than Earth's oceans, it might accumulate to higher concentrations because it's not being destroyed as fast.
That's one possibility. Another is that photochemical reactions in the hydrogen atmosphere could produce DMS from simpler precursors without any biology involved. Some researchers have run models showing this could happen.
What about the comet detection? You said DMS was found on Comet 67P?
Yes, by the Rosetta mission's mass spectrometer. This was a shock to astrobiologists because comets are essentially frozen balls of ice and rock. If DMS can form in a cometary environment, it can clearly form without life.
Does that completely invalidate DMS as a biosignature?
Not completely, but it complicates things. The researchers who made the comet detection argue that comets have a pristine inventory of organic molecules formed through abiotic processes in the early solar system, but the amounts are tiny compared to what's apparently on K2-18b.
So the argument becomes, yes, small amounts of DMS can form abiotically, but the huge amounts on K2-18b still require biology.
That's Madhusudan's position. He argues that the sheer quantity of DMS required to produce the spectral signal is so large that non-biological sources can't explain it. You'd need a massive, continuous flux of DMS to maintain those atmospheric concentrations.
And the counter-argument is?
That we don't understand the chemistry of hydrogen-rich ocean-world atmospheres well enough to make that claim. There might be feedback loops or reservoir effects we haven't modeled. The absence of evidence for abiotic DMS production isn't evidence of absence.
What about other biosignatures on K2-18b? Is DMS the only candidate?
The methane detection is actually interesting too. Methane is considered a potential biosignature because on Earth, most atmospheric methane comes from methanogenic microorganisms, methanogenic bacteria, and archaea. In fact, some researchers think the methane on K2-18b could indicate methanogenic life even without the DMS.
But methane can also form geologically, right?
It can. Volcanic activity, serpentinization of rocks, and other geological processes produce methane. So methane alone isn't a strong biosignature, but methane combined with carbon dioxide in a hydrogen atmosphere with low ammonia and potential DMS, that combination is harder to explain without invoking biology.
It's like building a circumstantial case.
Great analogy. No single piece of evidence is conclusive, but if multiple independent lines of evidence all point the same direction, the case becomes stronger.
What would it take to make a convincing case for life on K2-18b?
First, we need to definitively confirm the DMS detection at 5 sigma or higher. That requires more JWST observation time. Second, we need to rule out abiotic explanations, or at least show they're implausible given the observed concentrations. Third, we need multiple independent biosignatures that are all consistent with biological activity.
And even then?
Even then, some scientists would remain skeptical. The history of claimed biosignature detections is littered with false positives. The 2020 phosphine detection on Venus was initially heralded as a potential sign of life, but subsequent analysis showed the signal might have been misinterpreted, and even if phosphine is present, volcanic activity could explain it.
So the scientific community has been burned before.
Multiple times. There was the famous 1996 claim of fossilized bacteria in a Martian meteorite, ALH 84001. That made global headlines, but subsequent research showed the key evidence, magnetite crystals, could have formed through non-biological processes.
The extraordinary claims require extraordinary evidence standard.
Exactly. Claiming you've detected life on another planet is about as extraordinary as claims get. The evidence bar is correspondingly high.
Let me ask a philosophical question. If we're so worried about false positives, how will we ever be confident we've detected alien life?
That's the crux of the matter. I think it will require multiple lines of evidence that all converge on the same conclusion, independent verification by different research teams using different methods, and theoretical work showing no plausible abiotic explanation exists. Even then, there will probably be debate for years.
Years seems optimistic. People still argue about whether viruses are alive.
Fair point. But the K2-18b situation is actually pushing the field to develop better frameworks for evaluating biosignature claims. NASA has created a confidence of life detection scale that grades evidence from 1 to 7, with 7 being absolutely certain.
Where does K2-18b currently fall on that scale?
If the DMS detection is confirmed, probably somewhere around 3 or 4, potential biological signal detected, but abiotic explanations not yet ruled out. We would need much more work to climb higher.
So we're in the early stages of what could be a decades-long investigation.
Potentially. But here's the exciting part. The tools are getting better. JWST is just the beginning. The Habitable Worlds Observatory, planned for the 2040s, will be specifically designed to search for biosignatures on Earth-sized planets in habitable zones. It will directly image planets rather than relying on transit spectroscopy.
Will it be able to study K2-18b?
K2-18b will likely still be a target, though the focus will shift to smaller, more Earth-like planets. The irony is that by the time we have instruments powerful enough to definitively characterize K2-18b, we might have found even better candidates.
Or we might confirm that K2-18b really is the first place we found alien life.
That's the tantalizing possibility, and that's why this planet has captured so much imagination despite all the caveats.
All right, let's engage in some informed speculation. If there is life on K2-18b, what might it look like?
First, let me emphasize that we're firmly in speculation territory, but informed speculation based on what we know about biology and the conditions on K2-18b.
Understood. So, aliens?
If there's life, it's almost certainly microbial. We're talking single-celled organisms, not little green beings. The conditions that seem most plausible on K2-18b, a deep ocean under a hydrogen atmosphere, would support microbes similar to extremophiles on Earth.
Why only microbes?
Several reasons. First, the energy available in a Hycean environment is probably limited. Without an oxidizing atmosphere, you can't have the kind of energetic metabolism that supports complex multicellular life. On Earth, the evolution of animals required the Great Oxidation Event, when photosynthetic bacteria filled the atmosphere with oxygen.
And K2-18b's atmosphere is reducing, not oxidizing.
Right. Any life would be anaerobic, not requiring oxygen. On Earth, anaerobic life exists, but it's mostly microbial, bacteria and archaea that live off chemical gradients rather than oxygen.
What chemical gradients would be available on K2-18b?
That depends on the planet's geology. If there are hydrothermal vents at the ocean floor, where the ice mantle meets the rocky core, you could have chemical energy from hydrogen sulfide, methane, or other compounds seeping up from the interior. This is actually similar to how some of Earth's deep-sea ecosystems work.
The tube worms and bizarre creatures around black smokers.
Exactly. Those ecosystems don't rely on sunlight at all. They're powered by chemistry from the Earth's interior. A Hycean ocean could support similar ecosystems.
What about photosynthesis? Could life on K2-18b use light from its star?
Interesting question. K2-18 is a red dwarf, so its light is shifted toward the infrared compared to our sun. Any photosynthetic life would need to adapt to those wavelengths. On Earth, some bacteria can photosynthesize using infrared light, so it's not impossible.
But the ocean is probably deep, right? Light wouldn't penetrate very far.
Correct. If the ocean is hundreds of kilometers deep, the surface layers might receive enough starlight for photosynthesis, but most of the ocean would be pitch dark. Any life in the deep ocean would need to rely on chemosynthesis, deriving energy from chemical reactions rather than light.
So we might be looking at something like microbial mats floating near the surface and chemosynthetic communities in the deep.
That's one possibility. Another is life concentrated around hydrothermal vents on the ocean floor, similar to the ecosystems we find at mid-ocean ridges on Earth. These ecosystems can be remarkably diverse despite having no connection to sunlight.
If the life is microbial, how would it produce DMS?
On Earth, DMS is produced when certain compounds made by phytoplankton break down. The phytoplankton make a molecule called DMSP, dimethyl sulfoniopropanate, which helps them regulate osmotic pressure in salty water. When DMSP breaks down, it releases DMS.
So the DMS on K2-18b might be a byproduct of alien phytoplankton.
Possibly. Or it could be produced by a completely different biochemical pathway we haven't imagined. The key point is that DMS is a sulfur compound, and sulfur metabolism is thought to be one of the oldest forms of metabolism. Some researchers think the earliest life on Earth used sulfur-based chemistry.
So sulfur-based life might actually be more universal than oxygen-based life?
That's one hypothesis. On Earth, sulfur-metabolizing bacteria and archaea are found in some of the most ancient lineages. If life emerges wherever there's liquid water and chemical energy, sulfur metabolism might be a common feature.
What about the biology itself? Would alien microbes use DNA like Earth life?
That's a huge unknown. DNA is the information storage molecule for all life on Earth, which suggests it's a good solution to the problem of heredity, but we don't know if it's the only solution.
Could alien life use a completely different genetic system?
Theoretically, yes. Some researchers have explored alternative nucleic acids, molecules similar to DNA but with different chemical structures. RNA-based life is also possible. There's a hypothesis that early Earth life used RNA before DNA evolved.
What about the building blocks? Would alien life use the same amino acids as Earth life?
Another unknown. Earth life uses 20 standard amino acids to build proteins, but there are many more amino acids possible in chemistry. Some have been found in meteorites, suggesting they form naturally in space. Alien life might use a different set.
This is both exciting and frustrating. We have no idea what we're looking for.
That's actually a feature, not a bug. If we know exactly what alien life looked like, we'd be biased towards finding only that. The uncertainty forces us to think broadly and consider possibilities we might otherwise overlook.
But it also means we might not recognize alien life even if we're staring right at it.
That's a real concern. If K2-18b hosts life with a completely different biochemistry, the biosignatures might not be ones we've thought to look for. We're searching for DMS because we know it's associated with life on Earth. But what if K2-18b's life produces something we've never considered?
How do we solve that problem?
By looking for chemical disequilibrium. Life, regardless of its specific chemistry, tends to push its environment away from chemical equilibrium. If we see a mixture of gases that shouldn't coexist without something actively maintaining them, that's a hint that something interesting is happening.
Methane and oxygen together would be an example.
Perfect example. Methane and oxygen react pretty quickly with each other. Finding both in an atmosphere means something is producing at least one of them continuously. On Earth, life produces both.
On K2-18b, the combination of methane and low ammonia is the equivalent.
It's suggestive. In a hydrogen-rich atmosphere, ammonia should be stable. Its absence suggests something is removing it, possibly an ocean, possibly biology. Combined with methane and potential DMS, the picture becomes more intriguing.
Let me ask a provocative question. If we confirm life on K2-18b, what does that mean for the origin of life question?
If we find life on K2-18b, it would suggest life isn't unique to Earth, that the universe produces life relatively easily under the right conditions. This would have profound implications for the Drake equation and our estimates of how common intelligent life might be.
Because if life emerged independently on K2-18b, it probably emerged many other places too.
Exactly. The key word is independently. If K2-18b's life shares a common origin with Earth life, panspermia, for instance, that's interesting, but doesn't tell us much about how easy life is to create. But if it arose separately, that's revolutionary.
How would we distinguish between those scenarios?
By analyzing the biochemistry. If alien life uses DNA with the same genetic code as Earth life, that would suggest a common origin. If it uses a completely different system, that would suggest independent origins.
Can we do that analysis from 124 light-years away?
Not with current technology. We can detect biosignature gases, but we can't sequence alien DNA. That would require a sample, either from an interstellar mission or from alien life somehow reaching our solar system.
Interstellar mission to K2-18b?
Not happening in our lifetimes. At current spacecraft speeds, it would take hundreds of thousands of years. Even with advanced propulsion like light sails accelerated by lasers, we're talking centuries.
So we're stuck with remote detection for the foreseeable future.
For K2-18b, yes, but Mars, Europa, and Enceladus, those are within reach. If we find life in our own solar system, we can study its biochemistry directly.
And that would inform our interpretation of places like K2-18b.
Enormously. If we find life in our solar system shares Earth's biochemistry, it suggests panspermia might be real, life spreading between worlds on meteorites. If it's different, that suggests life emerges independently wherever conditions allow.
The next few decades are going to be wild for biology.
If we're lucky enough to make these discoveries, yes, astrobiology might become a truly experimental science rather than pure theory.
All right, let's zoom out and talk about the big picture. What does K2-18b mean for the search for extraterrestrial life?
K2-18b represents a paradigm shift. For decades, the search for life focused on finding Earth twins, rocky planets in the habitable zones of sun-like stars. K2-18b challenges that approach by suggesting habitable conditions might exist on planets completely unlike Earth.
The Hycean paradigm.
Exactly. If Hycean worlds can support life, the number of potentially habitable planets in the galaxy increases dramatically. Sub-Neptunes are far more common than Earth-sized rocky planets, and they're much easier to study spectroscopically.
Why easier to study?
Size and atmosphere. A planet like K2-18b has a radius 2.6 times Earth's and a thick, extended atmosphere. When it transits its star, it blocks more light, and its atmosphere intercepts more starlight, producing stronger spectral signatures. Earth-sized planets with thin atmospheres produce much weaker signals.
So the irony is that the best candidates for detecting biosignatures might not be Earth twins.
That's Madhusudan's whole point. He's argued that we might detect biosignatures on a Hycean world years or decades before we could detect them on an Earth twin, simply because the signals are stronger.
What about future missions and telescopes? What's coming that could advance this research?
In the near term, more JWST observations. K2-18b will definitely get additional telescope time to try reaching that 5 sigma threshold for DMS. Beyond that, we have the Ariel mission, the Atmospheric Remote Sensing Infrared Exoplanet Large Survey, launching in 2029. It's designed specifically to characterize exoplanet atmospheres.
Will Ariel be better than JWST for this?
Not necessarily better, but complementary. Ariel will observe many more planets, building up statistics on atmospheric compositions. JWST will continue doing deep dives on individual targets. Together, they'll give us a much broader picture of exoplanet diversity.
And the Habitable Worlds Observatory you mentioned?
That's the big one. HWO is NASA's flagship mission for the 2040s, specifically designed to search for biosignatures on potentially habitable exoplanets.
It'll directly image planets rather than relying on transits, which opens up a much larger sample of targets.
Right. With transit spectroscopy, we're limited to planets that happen to pass between us and their star. That's a small fraction of planets. Direct imaging can study planets at any orbital orientation, vastly expanding what we can observe.
That's exciting, but we're talking 2040s.
2 decades away. Good science takes time. The James Webb Space Telescope was first conceived in the 1990s and didn't launch until 2021. Major space telescopes are multi-generational projects.
What about ground-based telescopes?
The extremely large telescope under construction in Chile will have some capability to characterize exoplanet atmospheres when it comes online around 2028. Ground-based telescopes have to deal with Earth's atmosphere, which is challenging, but they can build much larger mirrors than anything we can launch into space.
So the next decade will see a steady stream of new data.
Absolutely. And here's what I find most exciting. We're entering an era where finding biosignatures is technically possible. Whether we actually find them depends on whether they're out there to be found, but for the first time in history, we can genuinely search.
Let's talk about the philosophical implications. If we do confirm life on K2-18b, how does that change humanity's perspective?
That's a question that goes far beyond science. Finding life elsewhere would be the most significant discovery in human history. It would fundamentally alter our understanding of our place in the universe.
Some people find that terrifying.
And some find it exhilarating. I think the reaction would depend heavily on what kind of life we find. Microbial life in a distant ocean is intellectually revolutionary, but doesn't pose any practical threat. It's not like we're going to have diplomatic relations with alien phytoplankton.
But it would raise questions about intelligent life.
Inevitably. If simple life emerges easily, does complex life? Does intelligent life? The Fermi paradox, where is everybody, becomes even more puzzling if we find the universe is teeming with microbial life, but seemingly devoid of technological civilizations.
Maybe life is easy, but intelligence is hard.
That's one possible resolution. Another is that intelligence emerges, but doesn't last long enough to spread across the galaxy. Or that intelligent life is common, but not interested in communication. We simply don't know.
Madhusudan had an interesting quote about this, something about whether humanity is prepared.
Yes, he said, "A bigger question in my mind is whether we as a species are prepared to find life as we don't know it. We as a society, as a species, should come together and ask ourselves, what is it that constitutes life elsewhere?"
That's profound. We might find something that challenges our very definition of life.
Exactly. Life on Earth follows certain patterns: cellular structure, DNA, metabolism. But those might be parochial features of Earth life, not universal requirements. Alien life might be so different that we struggle to even recognize it as alive.
How does that affect the search?
It means we need to cast a wide net. Look for chemical disequilibrium, unexpected combinations of gases, anything that suggests active chemistry that shouldn't persist without some driving force. Stay open to surprises.
Rapid-fire questions to wrap up. Ready?
Go for it.
What's the probability K2-18b actually has life?
Impossible to quantify meaningfully, but high enough to justify serious investigation.
When will we know definitively whether DMS is there?
If more JWST time is allocated, potentially within a few years, the 5 sigma threshold is achievable with 16 to 24 additional hours of observation.
Even if DMS is confirmed, will scientists agree it indicates life?
No. Scientists never unanimously agree on anything controversial, but a strong DMS detection would shift the debate from "is it there" to "what's producing it."
Could K2-18b host complex life?
Almost certainly not. Conditions favor microbial life at best. Complex life requires energy sources and evolutionary timescales that probably aren't available.
Will humans ever visit K2-18b?
Not with any technology we can currently envision. At 124 light-years, it's beyond our practical reach for centuries at minimum.
Best case scenario, what could we learn about K2-18b in the next decade?
Confirmed presence or absence of multiple biosignature gases, better constraints on the planet's structure and whether it's truly a Hycean world, and potentially a definitive answer on whether DMS is real.
Worst case scenario.
The signals remain ambiguous, funding for follow-up observations gets cut, and K2-18b becomes a historical footnote rather than a breakthrough.
Final question. What keeps you up at night about this?
Two things. First, the fear that we might miss something, interpret a real biosignature as noise, and never follow up. Second, the fear that we might claim a detection prematurely, have it debunked, and set back the field. The scientific community has to thread a needle between excessive caution and excessive enthusiasm.
And you think we're threading it correctly?
I think the debate around K2-18b is exactly what healthy science looks like. Claim, counterclaim, demand for more data, skepticism balanced with openness. It's messy and frustrating, but it's how we get to truth.
Well, folks, that's where we'll leave it. K2-18b, a planet 124 light-years away that challenges everything we thought we knew about habitability, possibly harboring the chemical signatures of alien life, or possibly just fooling us with exotic chemistry we don't understand yet.
Either way, the fact that we can even ask these questions is remarkable. A decade ago, detecting molecules in the atmosphere of a potentially habitable exoplanet was science fiction. Now, it's science.
Do remember, like and share this video for others to see, and also subscribe to our channel. And if you have questions or thoughts, we'd love to hear from you.
Thanks everyone for spending these minutes with us exploring one of the most fascinating worlds we've ever discovered.
I'm Marcus Chen.
And I'm Dr. Elena Vasquez. Keep looking up and wondering what might be looking back.